The Moss Report is brought to you by Mycolife, makers of the world's best mushroom extracts, including the Moss Method mushroom formula, available exclusively at mycolife.us. This podcast contains a number of scientific terms. For your convenience, we've included a glossary at the link in the description or scan the QR code on your screen. Hello, healers. My name is Ben, and this is the Moss Report. I'm here today with my father, author and founder of the Moss Report, Dr. Ralph W. Moss. And we're joined with our special guest, Professor Thomas N. Seyfried of Boston College. And Professor Seyfried is the author of a book, Cancer as a Metabolic Disease, as well as the head of the research department doing some of the leading work on the whole subject of cancer as a metabolic disease. Gentlemen, thank you very much for being with us. Professor Seyfried, good to see you again. Thanks for joining us.
Well, thanks, Ben. It's always great to be back and with your dad, Ralph. It's always a pleasure.
Thank you. Thank you, Tom. Wonderful to see you.
So I just want to sort of lay the groundwork for our audience who may not be familiar with this topic. The conventional thinking around cancer and its origin relates to what is known as the somatic mutation theory, which in essence means that there is a mutation inside of the cell that causes uncontrolled growth. And the theory that is being put forth, the metabolic and mitochondrial theory, as I understand it's being now referred to, is that the origin of cancer actually stems from a malfunction, if you will, of the metabolic system of the cell. Would you say that's a fair characterization?
Well, the metabolic system, of course, is the mitochondrion. There's now massive evidence presented that all major cancers have some defect in the number, structure, and function of the mitochondria, all of which creates an environment where mutations occur as a downstream effect of the damage to the regulation of energy metabolism. Otto Warburg was the first to discover the origin of cancer as a mitochondrial metabolic disease back in the 1920s and 30s. But certain new facts were not available to him at that time. And he also made certain assumptions that turned out to be incorrect. So the two key pieces of information that derailed the entire cancer industry thrust to manage cancer was the absence of certain critical information and misinterpretation of the data that they thought was accurate. And as the result of that, the entire cancer field ran off chasing genes, which is underpinning much of the pharmaceutical effort to manage cancer today, and many of the drugs that are being developed are based on the somatic mutation theory, which is a no longer credible theory. And that can account for why we have over 618,000 people a year dying in the United States, which comes to about 1,700 people per day or 70 people per hour. And this relentless death and suffering continues year in and year out as the result of focusing on a theory that is no longer credible.
Mm-hmm. And Otto Warburg, whom you mentioned, got the Nobel Prize for this discovery. It was that important.
He got a Nobel Prize for his discovery of the respiratory enzyme, cytochrome C.
I see.
So he was nominated many times for his work in cancer, but never received it. And that was a variety of reasons. Hitler determined that Jews should not get or any German for that matter. You know, as a Jew, why was he spared by Hitler? Hitler feared cancer as much as anyone did and spared Warburg. But Warburg was no fan of Hitler. In fact, Sam Apple's book, Ravenous, goes into a lot of the details on this backstory of Warburg. But he understood cancer better than anyone at that time. As I said, his failure to recognize that there is a second fermentable fuel, which is glutamine, and the mechanism for that, which is mitochondrial substrate-level phosphorylation, was not known to him, and we've clarified that. And his assumption that oxygen consumption was an accurate marker for oxidative phosphorylation was not correct, and we've corrected that in our two big new papers. So that sets a lot of the controversy and misinformation and misunderstanding arose from measurements that were not accurate or information that was not available at that time.
He attached himself to a sort of simplistic view of cancer, right, Tom? That basically, I mean, he was very dogmatic personality and brilliant. Albert St. George told me he was the most brilliant biochemist of the 20th century, but he didn't have the information you have.
I went back, and in fact, our new big paper that just came out of the Journal of Biomembranes and Bioenergetics goes into the deep dive of where these mistakes were made. And Sidney Winehouse at the time, who was head of the NIH and National Cancer Institute, in fact, even today, they're making the same errors. They're assuming that oxygen consumption is an accurate marker for ATP production through oxidative phosphorylation in cancer cells, which is wrong. And we showed evidence. We and others have shown that there's a disconnect. Cancer cells do take in oxygen, but it's not used predominantly for ATP. It's used to make reactive oxygen species, which are the radicals that cause the DNA damage in the nucleus.
So sort of the opposite of the healthy use of oxygen.
Yes. And when Warburg, actually, he relied on his former postdoc, Hans Krebs, another giant in the field of biology. Hans Krebs and his group clearly showed in rat liver slices that each mole of oxygen consumed was equal to 7 moles of ATP produced.
So we should explain ATP being the packets of energy that are normally produced on a cell.
It's the biochemical currency of energy, adenosine triphosphate, because it drives all bioenergetic processes. And without energy, nothing can survive. So you have to have some source of energy always. And Warburg was saying that most of the energy from cancer came from a fermentation metabolism where glucose would be fermented to lactic acid. And he could then estimate how much energy was being produced in a cancer cell by how much oxygen it consumed and by how much lactic acid it produced. We have gone on and shown that none of those measurements are accurate. They're estimates. And I said, where did he get the idea that 7 moles of ATP could be produced from 1 mole of oxygen consumed? And it came from the work of Hans Krebs. And Krebs is a giant in the field of biochemistry. So Warburg trained Krebs and felt that Krebs could never be mistaken because I trained him. And Krebs was not mistaken when it came to normal cells, but he was mistaken when it came to tumor cells. And Warburg used the same concept. He said, my controls are kidney slices and liver slices, and I monitor how much oxygen they consume, and I estimate a certain amount of ATP based on each mole of oxygen producing 7 moles of ATP. And then he made slices of tumor, and he said, see, they consume much less oxygen, and they produce a lot more lactic acid. But he was still assuming each mole of oxygen consumed by a tumor cell still produced 7 moles of ATP, even though it was consuming less oxygen. And it was making up for the deficit by fermenting lactic acid to produce 1 mole of ATP. So 1 mole of lactic acid produced equals 1 mole of ATP. 1 mole of oxygen consumed is 7 moles of ATP. And he was using those quantifications. And Sidney Winehouse came along and said, listen, some cancer cells consume just as much oxygen as a tumor cell. Therefore, tumor cells cannot have a problem in oxidative phosphorylation. And Warburg also had the same data, but he said, but the tumor cells are still fermenting, meaning their mitochondria or something is abnormal. What we have clearly shown in our own research quantitatively that oxygen consumption is not an accurate marker for ATP production in cancer cells. It is in normal cells, but not in a cancer cell. So that questions now all of these ideas about where the cancer is getting its energy. Not only that, we have now with our work and others that the production of lactic acid is also not an accurate marker for how much ATP is coming through the cytosol, the cytoplasm, because they have different pyruvate kinases, which is the payoff phase of the glycolytic pathway. And in normal cells, you have a pyruvate kinase 1, PK1, pyruvate kinase M1 isoform, where you do get 1 mole of ATP produced by 1 mole of lactic acid produced. But in cancer cells, you have a PKM2, which produces lactic acid without producing ATP. So that estimation is wrong, and we put that in the paper as well. So it was the quantification of where energy was coming from and the amount of energy that confused the entire cancer field for decades.
Yeah.
And therefore, everybody ran off chasing genes. And now, we've done so much research on that. The genes are absolutely impossible to be driving cancer because nuclear mitochondrial transfer experiments ruled them out. So you have to look at the data on all aspects. And what we've discovered that Warburg did not know about, nor still do most people because they don't read the literature, is that we have shown that the amino acid glutamine, which the field thought was respired,
Meaning
that it was glutamine respiration. You need glutamine was driving energy through oxidative phosphorylation based on energy consumption. Well, we have shown that the oxygen consumption is no longer an accurate marker. So we took away all oxygen and we took away all glucose. So we didn't get the interference from either oxphos or fermentation in the cytoplasm. Use glutamine alone as a fuel in the absence of oxygen and in the presence of cyanide, which completely shuts down oxidative phosphorylation.
Mm-hmm.
And we see continued energy production, ATP. Well, where the hell is the ATP coming from if there's no glucose and there's no oxygen? So we teamed up with Christos Xenopoulos, the world expert on mitochondrial substrate-level phosphorylation, which is a fermentation mechanism inside the mitochondrion itself. So this organelle can not only get energy through oxidation, respiration, but also fermentation. This is an evolutionarily conserved that organelle is an organism that has certain capabilities. So
that's its biological, its evolutionary origin, right?
Yes,
is probably
absolutely.
that the mitochondrion was incorporated into a bacterial cell, which essentially created the normal eukaryotic cell. That's the best theory, at least.
Yeah, no, that's the
standard theory.
That's the recognized explanation.
Yeah.
But the issue, you have to realize that organelle not only directs the entire energy metabolism inside the cell from oxidative phosphorylation, which is a highly efficient process, but also it can ferment. And that fuel for fermentation is not sugar, it's the amino acid glutamine. So we were showing, and then people knew for years that cancer cells have glutamine addiction. Yeah. They just didn't know the mechanism by which the glutamine was driving the dysregulated growth. And we now show it's a fermentation metabolism inside the mitochondrion itself. So in order to manage cancer, and we interrogated all other fuels to see is there anything that could replace glutamine as an alternative fuel, and none of them can do it. None of the other 20 amino acids. Asparagine, a little bit, glutamate, of course, but the transporters for these are minor, and the amounts of these fuels in the microenvironment are minor. So if you want to drive a raging beast, you have to have a very high throughput process, and the other amino acids are not there in any level that would drive the beast. So glutamine is there in massive amounts because it's the most abundant amino acid in our body. So it already has all of this available, and plenty of sugar, plenty of glutamine, and you have a fermentation metabolism in the cytoplasm and in the mitochondria. And we have shown the exhaust system. Don't forget, when you breathe energy like we are doing now in our conversation, every breath I bring in air which has oxygen, and I exhale CO2 and water. Those are the waste products of normal respiration. The cancer cells' waste products are lactic acid and succinic acid. Succinic acid is the waste product of glutamine fermentation. So, and all cancer cells are blowing out succinic acid like there's no tomorrow. So you know they're fermenting glutamine. If you target glucose alone, the cancer cells will upregulate glutamine metabolism. And if you target glutamine metabolism, they upregulate glucose metabolism. So the way you manage cancer is you must target both of those together simultaneously while transitioning the body over to nutritional ketosis, because the tumor cells can't burn ketones or fatty acids, because you need a good, strong mitochondria, healthy mitochondria to do that. And no cancer cell has a normal content or composition or function of mitochondria. So the ketones keep the rest of our body healthy, our normal cells, non-tumorigenic cells, pick up fatty acids and ketones. Cancer cells are not able to do that. Therefore, they become marginalized and die when you take away their glucose and glutamine. The interesting thing is we have all the science to support this. We've done all the experiments. We have all the science and evidence to support this in both preclinical studies as well as some clinical studies. And almost nobody knows about it. And there's not a single clinical trial anywhere in the world doing what I just told you to do. And when you try, and if you did that, you would be able to manage cancer without toxicity for the majority of people. You just get a low glucose ketone index. And we found out these parasite drugs like mebendazole, fenbendazole, they not only target the glycolysis pathway, they target the glutaminolysis pathway. So when you do that under nutritional ketosis, these simple, cheap-ass parasite drugs, you know why? The parasites and the cancer cells are using the same mitochondrial substrate-level phosphorylation. Not complicated at all. And it works. It works really, really well. Not perfected yet, but it certainly shows tremendous hope without toxicity. So, I mean, what's wrong with this? It's just, and it's not that it's so simple, but it's not nearly as complicated as people made it out to be.
How is it that this eluded you when you wrote Cancer as a Metabolic Disease, this latest finding? I mean, it seems like so obvious now that the way you state it.
I speculated on it in chapter 8 of my book. It was a chapter of speculation.
Oh.
And I took the word from Dr. Hokachka, who has since passed away, when he was talking about animals that are held in hypoxic, they were like seals and porpoises and things. He held them underwater, and because they naturally could be underwater, but you can't do those experiments today. He was doing them in Hawaii. And he was measuring lactic acid and succinic acid in the bloodstream of these animals that were held underwater. And he was finding a tremendous amount of succinic acid increased in the bloodstream along with lactic acid. He predicted, based on his biochemical measurements, that there was a amino acid fermentation that was going on to keep these animals alive. The problem is he never speculated on what was the amino acid that was responsible for this. We now have shown that it's glutamine. And when we did a deeper dive on this, and the heart attack people in the cardiovascular have known about this for decades, it's just that the cancer world and the cardiovascular world don't overlap. So when people have heart attacks, their bloodstream fills up with lactic acid and succinic acid, because when you die from a heart attack, it's neurons that die. Neurons and cardiomyocytes, which rarely, if ever, become cancer, cannot shift to a fermentation metabolism for very long. So they can do it for a short period of time, a giant burst of fermentation, because if you don't, you're going to be dead. And you see a massive amount of succinic acid and lactic acid build up in the bloodstream of a person under a heart attack because they're not breathing. What they're doing is they're fermenting glucose and glutamine, and the waste products are lactic acid and succinic acid. And cancer cells are doing that even in the presence of oxygen. 100% oxygen, they're throwing out those two fermentable fuels. Warburg always said that was the unique thing about cancer. They continue to ferment in the presence of oxygen. And he was only looking at the lactic acid part of it. He wasn't looking at the glutamine producing succinic acid part. The cardio field provided us with the direct evidence for that. And then when we did our experiments, we designed a novel series of experiments to exclude oxphos and exclude glycolysis as the explanation. And we still got ATP in the absence of oxygen. And it was coming from the fermentation of glutamine, because we labeled the glutamine in a C13 labeled glutamine, and we got C13 labeled succinic acid, knowing exactly where it was coming from. And when we targeted the glutaminolysis pathway, we killed the cells instantly. So
You sort of clinched the argument there, right?
Yeah.
I mean, because you
Oh,
have
yeah.
It's right when you say labeled, you mean you made it radioactive, right?
yeah, well, we use radio labeled. Well, we can pick it up by mass spectrometry. We can know where the labels. So we add a labeled glutamine, and then we look for the waste product, and we found labeled succin in the same carbon. So you know where it came from, and that's what cancer cells are doing. They're dumping out acidic products that have carbon hydrogen bonds. So this is why you have to wasting the sun's energy. See, oxidative phosphorylation is so energetically efficient. Every carbon hydrogen bond is broken down, and the energy of the sun is captured in the carbon hydrogen bonds. So if you're dumping lactic acid and succinic acid, which have carbon hydrogen bonds, this is not an efficient. This is a primitive fermentation mechanism that existed before oxygen came into the atmosphere 2.5 billion years ago. So these cancer cells are doing nothing more than falling back on these ancient fermentation pathways. And you have to have high throughput, because in order to make up the loss of efficient oxidative phosphorylation, you better have a tremendous logistical supply of the fermentation fuels. That's glucose and glutamine are the originating fuels for carbohydrate fermentation, sugar fermentation, and amino acid fermentation.
Glucose is also the fuel for metabolism in the presence of
Oh, yes.
oxygen.
Yeah, right.
What you called oxphos,
Yes,
oxidative
yes.
phosphorylation.
Yeah, absolutely, because the pyruvate is then metabolized to acetyl-CoA, and you produce reducing equivalents. You're breaking down all of that in the TCA cycle, delivering the reducing equivalents to the electron transport chain, producing a very high proton motive gradient and generating highly efficient energy with water and CO2 as your waste product, CO2 coming out of the TCA cycle, and water being produced as the product through the F1, F0 ATPase. Highly, highly efficient mechanism. If that mechanism doesn't work well, you have to fall back on an ancient fermentation pathway. And that's what Warburg said. It's a two-step process. It's a chronic damage or disruption of efficient oxidative phosphorylation that is coupled to a compensatory substrate level, which we call it fermentation, which was now substrate level phosphorylation, which happens in the cytoplasm in the 10-step Embden-Meyerhof pathway, or in the mitochondria, which nobody knew about, which is a mitochondrial substrate level phosphorylation. But in chapter 8 of my book, I speculated. But then I reached out since that time to Christos Giannopoulos at Semmelweis University, who is the world leader on understanding substrate level phosphorylation.
In Budapest.
In Budapest.
Mm-hmm.
And he looked at my diagram. He says, no, that's not right. So, but he said, your idea is really exciting, but he said, the pathway you have is not right. So we then, over the last, 15 years, designed experiments to get to the right pathway and then figuring it out. And now we have, and we published it in ASN Neuro, which has now got the most views of any article ever published in that journal. And they still scratch their heads. They still have no clue. When I say they, I think it's the cancer establishment. They never heard of like what I'm talking about. Yeah. Substrate level phosphorylation in the mitochondria. What the hell is that?
What does that have to do with cancer?
Yeah, everything. It's the missing link in Warburg's central theory. Warburg was correct. He just didn't have all the stuff put together. So we can take the whole cancer field and put it back on the right track.
Ironically, though, you're also sort of confirming Sidney Winehouse and Warburg's other opponents, because in effect their criticism was a correct one, if I'm understanding you. I mean,
well, they didn't believe Warburg's explanation for it.
Right.
Winehouse was saying, see, if cancer cells consume oxygen at the same rate as normal cells, how could you say in any way that they have a problem in oxidative phosphorylation?
Yeah.
So they must use fermentation as like a turbocharge. They need so much energy. They need both oxphos and fermentation together. So, but I showed that's not correct, because the cancer cell does consume oxygen, but it's not producing very much. It does produce some, but it's neither necessary nor sufficient for the dysregulated cell growth. So cancer cells can get energy from three sources: the cytoplasm through the Embden-Meyerhoff-Barnes pathway, which is the producing ATP at the payoff phase of the glycolytic pathway. That's true. But we can't know for sure how much, but they do get some energy, a lot of energy from there, but we don't know exactly how much. Yes, they can get some energy from oxphos, but it's neither necessary nor sufficient in the absence of a fermentation metabolism. And yes, they get energy through the mitochondria from fermenting glutamine. So it's glutamine to glutamate to alpha ketoglutarate in the TCA cycle to succinyl-CoA, and then the succinate leaves the TCA cycle and is dumped out into the cytoplasm and into the outside the cell. So and that creates the acidification. When people say, "Oh, we can't kill cancer cells with radiation and chemo and immunotherapies because their microenvironment is so acidified," well, that's the crap products of the fermentation metabolism that acidifies. So if you take away glucose and glutamine, you remove the acidification of the microenvironment, now making these cancer cells super vulnerable to all these standards of care that people are. You don't have to use such high dosages. You don't have to be irradiating and poisoning people with the same level of toxicity. You can use those same procedures at much, much lower dosages and timing, because the cancer cells will now be super vulnerable. They're hanging on barely to survival without their two fermentable fuels. And now you just come in and push them over the edge with the standards of care that you have. And then that will allow cancer patients to eliminate their cancer far more effectively with minimal local collateral damage. What's wrong with that? I don't see anything wrong with this strategy. It's hard science that supports everything that I'm saying.
Well, the last time we spoke, Fen and Ben, these anti-parasitical drugs were not on the radar, if I remember correctly.
Yeah.
So that's something new, and they're very controversial because of people advocating them for COVID and other things.
Yeah.
But also, ivermectin appeared on the scene, and ivermectin is also, if I'm not mistaken, anti-parasitical. So there seems to be a sort of a trend here. You know, it seems like maybe that's because I think when we last spoke, Tom, the big hang-up was for me, and also many other people, was that it was fairly straightforward. It seemed fairly straightforward to reduce your glucose. A person can do that at home, and you've invented a way for them to measure how much glucose they have vis-a-vis ketones. The Glucose Ketone Index, right?
Yeah.
And so that seemed straightforward. What seemed much less straightforward was how do you control the flow of glutamine, being the most abundant free amino acid in the body, and also since it's not an essential amino acid, you can make it from other amino acids. How do you really get a handle on that? And now you seem to have indicated that this fenbendazole and mebendazole, which have been advocated just empirically over the years by that clinic in London and others, as a way of controlling that. Do you feel confident in that as a mechanism, or are you still looking for something more of a knockout punch?
Well, I think that's they're not as powerful as 6-deoxynorlucine, which is DON.
Yeah,
DON is the big stick on this whole thing. Mm-hmm. We published a paper in 2019 using a calorie-restricted ketogenic diet with low-dose DON for brain cancer, and it was really remarkable how powerful it is. And what we did is we showed that under ketosis, you can deliver DON through the blood-brain barrier 3 times more effectively, which is really powerful. Mm-hmm. And with embendazole and fenbendazole, people recommend taking those drugs with a fat diet, which means that basically it's a ketogenic. So you're facilitating the delivery of that. But in our paper that we have under review, right, we put it out in bioarchives. It was the first evidence that mebendazole is targeting the glutaminolysis pathway. People say that it targets microtubule assembly. Um,
Mm.
but there's a terrible toxic vincristine, I think, is the chemo drug that targets microtubules. And it,
Taxol too, I think.
yeah, and they're very, very toxic drugs.
Mm-hmm.
And
Right.
bendazole is extremely non-toxic. People take it to kill parasites, and it's very
Mm-hmm.
little toxicity.
Mm-hmm.
And yet it's very powerful against cancer. Um, and microtubule assembly requires ATP, and if you're blocking ATP, then you ain't going to get microtubule assembly.
Yeah.
So everything comes back to energy. So if we can, so the field was led down a path thinking that mebendazole was working and through microtubule assembly, but it might do that, but it's working more by targeting glycolysis and glutaminolysis pathways.
Yeah.
And most of that work was done in China and India because they have big parasite problems. So
Mm-hmm.
they showed that mebendazole blocked glucose transporters, so that would facilitate shutting down glucose. And we're the first ones to show that it reduces the activity of glutaminase, one of the top the enzymes that breaks glutamine down to glutamate. So that would put some pressure on the glutaminolysis pathway. And then we realized when I did a dive on the parasites, they used mitochondrial substrate-level phosphorylation, just like cancer cells do.
Mm-hmm.
So, and you're right, Ralph. When I started this, the political firestorm about ivermectin was out of control, and I didn't want to do anything with ivermectin because whatever I found would be would be
Right.
yelling and screaming about it.
Right.
So we went into mebendazole, which is an alternative, but I have not looked at ivermectin, and if ivermectin targets glucose and glutamine, then it can explain why it's so powerful as a therapeutic. But again, they don't work by themselves. You really need to put the patient in nutritional ketosis first, and then you come in with the parasite medications or DON. But that's why I developed the press pulse therapeutic strategy for the effective management of cancer in people, because we pulse glutamine targeting drugs. Because if you are too aggressive, you can harm your gut and immune system, and that's why we pulse these drugs. So the patient must first get into nutritional ketosis using the glucose ketone index calculator.
Yeah.
So once you're in low GKI, then you can come in with various parasite medications, which will then target these tumor cells. And we also showed that this drug, CPI 613, which targets pathway in the TCA cycle, it was very toxic when given to humans in pancreatic cancer, very toxic. And we gave it to the mice the same way, terribly toxic. And then when we administered it with the ketogenic diet, it became far, far less toxic. So clearly, we're able to rescue certain drugs that were considered unusable in the clinic. We think we can resurrect a lot of these cancer drugs by just simply putting patients into nutritional ketosis and then strategically pulsing these drugs using low dosages. Dosage timing and scheduling have to be worked out to maximize the therapeutic efficacy of each drug. But they will all work really well when the patient is in nutritional ketosis, dictated by the number generated from the glucose ketone index. So we have a strategy, a plan that once adapted, and it will drop cancer death rates massively and keep people alive with a higher quality of life much, much longer than would be predicted. So we're all at the very beginning of this. The science is solid. The practical application of this in the clinic needs to be worked out and improved for sure. But all the parts of the puzzle are there. We know what to do. We think we know how to do it. We try it on small case reports, clinical, not big ones, because nobody knows how to do this. If you take 100 patients and try to get them, it's like herding cats. Everybody has to be a little different than the other guy, and
Yeah.
how do you know all this? So we do it on small groups of people to learn what we need to do and how to do it, and the ones that are most successful, what did they do and how did they do it relative to those guys that aren't so successful. And then we keep logging this and logging it and putting it into apps and things like this, so that when we go and do a larger clinical trial, we'll be able to identify the variables that need to be regulated for each person, whether what their age is, what their sex is, dietary issues, all these kinds of things have to be corrected or analyzed or adjusted depending on the situation. So we can't run in and do, oh, where are your big clinical trials? Well, who the hell is going to do a big clinical trial when you have no clue what the hell we're doing? So you have to have some knowledge base. You have
Yeah.
to have some experience. And once that experience and knowledge base comes to pass, and we develop these tools and ways to do it, that's the major breakthrough in this whole cancer thing. You're going to start seeing people live far, far longer with a higher quality of life. There's no question, because the science tells us that. We have interrogated the. We know what to do, and basically how to do it. We just need to get the practical application in the clinic, and we just started a new society of International Society of Metabolic Oncology, where we're gaining interest by oncologists throughout the world who are chomping at the bit to treat their patients using these procedures, because they know that it will be work. And a lot of them have seen it in small groups of people. That, wow, how can you be pulling people out of hospice with just changing their diet, putting them into nutritional ketosis, and giving them a parasite medication? What the hell is going on with that?
But also, it isn't, it isn't quite so simple to get somebody into ketosis and then keep them there over a prolonged period of time. I speak from personal experience that, it gets, there are difficulties, lifestyle difficulties. So
Well,
it makes perfect sense as far as I'm concerned in terms of the science of it, but it's a good idea to go a little slow
Yeah.
at first, and then you have to educate the educators. You have to educate the people who are going to help the guides who are going to help the people get and stay in ketosis, and it's
Yeah,
complicated.
that's one of the challenges. And our Greek colleagues who had really remarkable results on glioblastoma just came out this year, where they
A
were,
very tough kind of brain cancer. People
yeah,
should
really
know. Yeah.
bad. But the ones that stuck to the calorie restricted Mediterranean diet lived 3 years or longer, were those that didn't. Only one person survived that long. And what they did was what did they do? They ate salmon, sardines, avocados, olive oil. I mean,
Yeah.
this doesn't sound like prohibitively difficult to do. And we're learning now. You can switch to carnivores. There's a lot of flexibility in keeping your GKI down without too much emphasis. Vegans seem to have a lot of trouble getting down, but they can. But it's more challenging for them. But people who are omnivoric and they can go to calorie restricted, and then the exercise is unbelievable. Even if people they say, oh, I'm GKI, I've been done everything five, I'm a five, I can't get down to your two level. I'm doing this and I'm doing that. I said, go out and walk, go out and run 4 or 5 miles a day. All of a sudden, they're down in the zone because your muscle activity is sucking the glucose out of the bloodstream, giving you a lower GKI. And then you start with the carnivore and the salmon and the olive oil and all this kind of stuff. It's not perfectly easy. Okay, you're not eating jelly-filled donuts and you're not going to the rice and bread and all this kind of stuff. If that's what you consider the greatest challenge in this, then you'll never achieve the goal of managing your cancer effectively.
Well, correct me if I'm wrong, but the body does store a tremendous supply of glutamine that it can tap into, that can sort of be accessed over a period of weeks
Yeah.
and months. So even if you cut off your immediate simple sugar intake, you still got fuel for the cancer.
Yeah, well, that's why you need the drugs. That's why you need the parasite medications and DON. That's why you need specific drugs to target the glutamine. That's why you're right. You can't. There's no diet that will lower glutamine, no matter what you do.
Right.
Cahill and his group down here at the Joslin Diabetes Center were able to show that the only thing that lowered glutamine was massive exercise, and it never lowered it to the level that would kill tumor cells. The level we have is so much above the threshold for survival for a tumor cell, the availability of the glutamine. That's why you need specific drugs to target that glutaminolysis pathway. But that's where the knowledge base has to come in. You need to know dosage timing and scheduling for those drugs so you don't harm and damage the gut or the immune system of the patient.
So are you saying that even if you went on a low carb diet, keto diet, and were in ketosis and kept it there for a few months, let's say, that even that's not really going to significantly lower the glutamine levels available in the body to the point where you're actively able to sort of starve the cancer of the glutamine that it's looking for?
For some people, that's true. For other people, it's not, because we're seeing people just getting into really low GKI values and no evidence of disease, which to me is like, well, what the hell? Why are they not using the glutamine? And then we see other people who do get into low GKI, and the cancer continues to be persistent, but we've never targeted the glutamine. In our preclinical systems, when we target the two together, is when you really get the power. And the mice are not always dying from cancer; they're dying from the adverse effects of long-term targeting of glutamine. So that's why we need to learn how to pulse the glutamine. That's going to be the clinical contribution to this whole thing. People are going to know what age you are, what sex you are, what's your cultural background, what's your religion, what are all these kinds of things have to come into how you're going to be pulsing the glutamine problem. But we know what we need to do. It's perfecting the strategy for doing it, which is now clearly achievable. If you think cancer is a genetic disease, none of what I'm talking about is achievable. So there, by the grace of God, you survive one of these toxic things. And then if you do survive using standards of care the way we have it today, you often suffer from lifelong health issues as the result of your body being brutalized, either surgically mutilated or poisoned or irradiated, that you're rarely ever the health that you were before you started all of this stuff. But with metabolic therapy, not only can you manage the cancer, but you can achieve a new level of health status that you never had before. Because once you start burning ketones, it's a super fuel. We evolved to burn ketones. We didn't have, as my late friend Bud Veach would say, we didn't have delicatessens on every corner during the Paleolithic period. Put us at risk for all the problems that we have today. So we're just throwing people back into a more Paleolithic diet and lifestyle, and then coming in strategically with drugs that will target the very difficult part of the glutaminolysis pathway. We have not yet found any tumor that can survive for very long without glucose and glutamine. So clearly, there's nothing else that can replace these two fuels. And they're not available in the environment in abundant enough levels. Glutamine is the only amino acid where no energy is invested to get energy.
Wow.
So it's gold. All other amino acids have to be reconfigured to get to alpha-ketoglutarate in the TCA cycles. There's an energy utilization to get energy from the molecule. So it's kind of a sum game. You use energy to get energy. You don't really benefit. Whereas glutamine is pure energy, and also it delivers the nitrogen needed for DNA and RNA synthesis and amino acid synthesis. So the nitrogen is coming from the glutamine, as well as the energy. Whereas the metabolites for fatty acids and all others are coming from the carbons of glucose. So you got the carbons of glucose, the carbons and nitrogen of glutamine, and we showed in our paper the powerful synergy of the overlapping metabolitic pathways of these two fuels driving the dysregulated cell growth. Yeah. Incredible. It's absolutely incredible.
The question that's going to be on many people's mind is the disconnect between the logic of what you're presenting and the factual nature of what you're saying and the demonstrable nature of it. After so many years that you've put into this, why this is still even unknown to the vast majority of people in the field or facilely dismissed. So maybe you could address that.
That's what happens whenever you have a paradigm shift of this magnitude. You get the same response. Confirmation bias in the cancer field is so powerful, which is just, it can't be right because everybody's doing something different. If you're right, how come 99% of people are doing, and they never actually look at the data about that. And dogmatic ideology is unbelievable. It's like a religion. It's very hard to convince somebody to do something different when their salary depends on them not doing it. That was Upton Sinclair said that.
Oh, right,
And if you
right,
and if you
right,
and if
right.
you look at Tolstoy, I have these quotes.
Yeah.
It's easy to get somebody who doesn't have like a dog in the fight to understand what you're doing, but it's almost impossible to convince somebody who is in the fight to change what they think.
Yeah.
So you have these confirmation biases. The National Cancer Institute on their website says cancer is a genetic disease, 100 different diseases. And they award grants for people hunting for these genes, the ever elusive cancer gene. And they keep doing it over and over. If you're at a top medical school and you need to justify your existence, you need to get grants. And those grants say cancer is a genetic disease driven by mutations. So you're going to be hunting for mutations only because the system tells you to do that. And if you choose not to do that, you lose your job. So you can look at all you want. But I went back and I looked at these paradigm changes, and the heliocentric geocentric is a really good example. For 1,800 years, Aristotle and Ptolemy were arguing that the Earth was the center of the solar system, and they kept doing more equants, epicycles, and deference, and all these kinds of things. And all the mathematicians of the time, the astronomers, were all trying to figure out where these planets were. How could you predict where one planet would be at any time of the year, and the movements of the celestial bodies? And it was a complete mess for 1,800 years. And Copernicus comes along and says, what happens if we replace the Earth with the sun? And of course, the Catholic Church went nuts. And there was they didn't accept that until like the 1800s.
Right.
That the sun was the center of the solar system, and people who challenged them were put in jail or house. Look at Galileo, house arrested. And then you have to look at the difficulty that Louis Pasteur had to go through with his germ theory. Pasteur took a beating. And so did Darwin Wallace on the theory of evolution, which still is under attack all the time. So the mitochondrial metabolic theory replacing the somatic mutation theory is the next great paradigm shift in the field of medicine. No question about it. And what we have found is it's not just cancer. The GKI can be effective in managing all major chronic diseases. You go up and down the list: diabetes, cardiovascular disease, dementia. These things are all aspects of mitochondrial dysfunction. Cancer just happens to be a capability where mitochondrial dysfunction is compensated for by substrate-level phosphorylation, where some of these others are not compensated and you get dead cells. So you can look back and see this whole thing. This is happening. The mitochondrial metabolic theory will replace the somatic mutation theory and all other theories, because it's the only theory that it can account best for the facts of the disease. It's just a matter of time, and it's confirmation bias, dogmatic ideology, and your paycheck that are pretty much standing in the way of this transformation. And people want it. It's not like we're dragging cancer patients kicking and screaming into this. They want it. They want to know they can manage their disease without toxicity. I mean, it's people's choice. You're allowing an individual to make decisions about their destiny. And there's power in that, and people fear the surgical mutilations and the toxicity of what they have to go through as much as they fear the disease. And that puts the fear of God, all this emotional distress, and this is not necessary. When you look at cancer as a mitochondrial metabolic disease, this is an opportunity to retool. To recharge your existence, come out of this better than you went into it. So, and because it's there, we have it. It's just that it has to be perfected. That's all. We're in contact with people who are doing this, and they're feeding us back massive information about what they've experienced and how and the doctors are always miffed. No evidence of these. What did you do? And a lot of them are not even curious. The oncologist, oh, wow, that's unbelievable. I never saw anything like this before. Don't you want to know? No. Just keep doing what you're doing.
Right.
Give me a break. The whole system needs to be reevaluated.
Yeah.
And it's going to happen. I mean, we're working on breast cancer, we're on pancreatic cancer besides glioblastoma. So we're going right through the list. And we're showing that every one of these cancers, they all have a very similar metabolic problem. They can't survive without glucose and glutamine, and they can't transition to fatty acids and ketone bodies, regardless of what you might read. We interrogated the damn cells. They can't. You take away glucose and glutamine, they're not going to be able to switch to any other fuel and survive. Yeah. I don't give a damn what anybody says. This is a mitochondrial metabolic disease, absolutely a mitochondrial metabolic disease, which now opens a whole new world of way to attack this problem. But they're hanging on to that theory. We just read a paper not long ago where they screened 200 million mutations looking for the singular driver that we've overlooked that's really responsible for this, and they didn't find it. And rather than concluding that, we'll never find it, they said, the search must continue. I mean, what do you have? How long you have to beat this dead horse before you're not going to get up and walk? Because there are millions and millions of dollars invested in finding that driver gene that no one's been able to find.
Right.
It does not exist. Get over it. It's the mitochondria, stupid.
So, Professor, you've described that you're still looking into the different cancer types and applying your theories to those cancer types to sort of bear them out. Is there anything other than that that's on your horizon for your lab right now?
Well, we're trying to find glutamine targeting drugs and neutralize any level of toxicity. You want to maintain therapeutic efficacy without collateral toxicity. So if we can figure out how to do that, we'll have a whole range of new drugs that we can use to manage cancer. We just have to figure out that. And all of our research is supported by philanthropy and private foundations. And that drives what we do. There are people out there that just want to see this paradigm change happen, and they don't feel they got to make $1 billion on it. And what our research is doing is, again, perfecting dosage, timing, and scheduling of diet drug cocktails that will ultimately be utilized in the clinic. It's always good to know that you've tried something in a preclinical system before you try it on people for the first time. It gives both the patient and the caregiver some confidence, peace of mind that you're not going to kill your patient. So, we've tested it and we've seen therapeutic success. And therefore, if you guys do it to some level the similar way, you should get the same result. You have to realize when we do stuff in mice, the basal metabolic rate of our experimental system is 7-fold faster than that of a human. So we always have to be cognizant of differences in basal metabolic rates. So then humans do much better than these mice because we can ask humans how they feel, and it takes a longer period of time for seeing therapeutic efficacy or toxicity, whereas in the mouse, everything happens so much faster. So we're able to do that, and how to translate from the research bench to the clinic is we're one of the few groups that actually do this. There's a lot of talk about doing it. Yeah. We do it. We have an army of clinicians out there that are chomping at the bit and trying it on these patients, and they feed it back to us, and then we strategize with our preclinical systems. So it's a feed-forward feedback system, and it's going to eventually solve this cancer problem.
Mm-hmm.
Great.
Well, kudos to you and to your lab associates and to those who keep you guys funded for all the work that you're doing. We, of course, are going to continue to follow the developments that you publish with great interest and with our appreciation.
Well, thank you very much. You guys are doing your fair share of pushing this system as well.
Thank you. It's been a highlight of my career to know you and to visit with you. And I keep eagerly following your work and keeping an eye on it. So congratulations, and look forward to even greater levels of cooperation.
Oh, thank you very much, Ralph. It's a real pleasure.
Thank you.
And Ben, you enjoy yourself now. Have a nice day.
All right, thanks. For now, for the Moss Report, I'm Ben Moss. How are you healing today?